Harold Scherhausen, born March 11, 1936, in Gelsenkirchen, Germany. As a child, Hausen experienced the Second World War in Gelsenkirchen Burr, which was heavily bombed. Fortunately, all of his family members survived. Schools were closed due to bombing raids in 1943, so Hausen's elementary education was full of gaps. When he re-entered school at the age of 10 in 1946, his lack of education was evident, causing him to struggle during the first year. In 1950, his parents moved to northern Germany. There, Hausen finished high school in 1955 with Abitur. In 1955, Hausen also briefly considered whether to study biology or medicine. Although his great interest for biology, he opted for medicine and started his studies at the University of Bonn. Hausen's first 2 years were particularly hard because he decided to attend lectures and courses in biology as well. His first exam after 5 semesters was passed with good grades. This made him confident for the semesters to come, which he spent at the University of Hamburg for 1 year and at the University of Dusseldorf, where he graduated in medicine and finished his MD thesis in 1960. Hausen received an offer from Werner and Gertrude Henle at the Children's Hospital of Philadelphia. Werner Henle headed the Division of Virology. Hausen accepted the offer at the end of 1965. The Henle lab was deeply interested in the newly discovered EBV. EBV is also called the human herpes virus 4 and is one of the 8 human viruses in the herpes family, and it is also one of the most common viruses in humans. It is best known as the cause of infections in mononucleases and is associated with particular forms of cancer. Harold moved to Philadelphia alongside his family. Hausen urged Werner Henle to permit him to work with a different viral antigen with the rest of his team. Reluctantly, Henle agreed.
He began to work eagerly on the introduction of specific chromosome aberrations in adenotype 12 infected human cells, as well as studying the DNA replication disturbance of individual chromosomes in human lymphoblastoid and lymphoma cell lines.
To prove to his mentor that his experiment worked, he had to microscopically show the presence of EBV particles in individual antigen-positive lymphocells. By doing this experiment, Hausen found out that the immortalizing function of EBV was being demonstrated in human B lymphocytes, and also discovered that the virus was a causative agent in infectious mononucleosis. So basically, the adenotype virus 12 infected human cells with EBV so they could diagnose infectious mononucleosis. In 1968, Hausen received an offer to go work at a newly opened Institute of Virology at the University of Würzburg, Germany. He was offered an establishment of his own with an independent group and granted support for the quick start in the German academic system. He accepted the offer in March of 1969.
His intention was to prove that EBV DNA persists in every tumor cell of Burkitt's lymphoma and that it does not establish a persistent infection.
Harold received a large amount of lymphoma cell lines and tumor biopsies from Warner Henley and George Klein. These biopsies included material from nasopharyngeal carcinomas.
Nasopharyngeal carcinoma is the most common cancer originating in the nasopharynx, which is the uppermost region of the pharynx.
By the end of 1969, we had the first data available that the non-EBV producing Burkitt's lymphoma cell line, Raji, contained multiple copies per cell of EBV DNA.
In 1974, Hausen was appointed chairman of the newly established Institute of Clinical Virology.
You've been doing a great job, and we're wondering if you were interested in the chairman position for the institute.
Yes, of course.
All right, but you have to change your focus on.
He had to change his main focus to cervical cancer.
All right.
Since Hausen's previous work in EBV led to the identification of EBV DNA in specific human cancers, he tried to use the same technique for SHV2 sequences in cervical cancer biopsies. However, all of his attempts failed.
HSV2 is also known as human herpes virus and causes most cases of genital herpes. This is very contagious and is spread when an infected person is producing and shedding the virus.
In late 1979, Harold and his co-workers Lutz Gisman and Ethel Michael de Villar successfully isolated and cloned the first DNA pruned genital warts, HPV-6. This DNA was also helpful in isolating another closely related genital wart, papillomavirus or HPV-11.
Yes, we did it. Clone them. By using the HPV-11 as a probe, 1 out of 24 cervical biopsies turned out to be positive, and in other biopsies, some faint bands became visible, permitting the speculation that they might represent hints of the presence of related, but different HPV types in these cancers.
In 1983, Hausen and his team were able to document the isolation of HPV-16, and in 1984, the isolation of HPV-18 DNA.
We noted from the beginning that HPV-16 DNA was present in about 50% of cervical cancer biopsies. HPV-18 was slightly more than 20%, including cervical cell lines, among them the HeLa cell line.
Within the first 2 years after isolating HPV 16 and 18, it became clear that these viruses must play an important role in cervical cancer development because viral DNA was commonly found in an integrated state, indicating the clonality of the tumor, and part of the viral genome frequently became deleted in the process of integration. Also, 2 viral genes, E6 and E7, were consistently transcribed in the cancer cells. Precursor lesions of the cervical cancer also contain these viruses and express the respective genes.
The early hypothesis that cervical cancer was caused by HPV and the successful isolation and characterization of the 2 most frequent HPV types in this cancer and subsequent steps leading to a better understanding of the mechanism of the preventive vaccine were cited as the prime reasons for awarding 1/2 of the Nobel Prize for Medicine or Physiology to Harold Hausen in 2008.
Wait, hold up. All right.
You're cheating.
Chill. You can...
What do I do? What do you want? What are you following?
You said...
Action.
He began to work eagerly on the
Behind the scenes.
Maybe I can buy
We did it. Yeah.